Hello, so my name is Paul Delaney,
My name is Cassie Tamiko.
and today we're talking about endo-1,4-xylanase. And today I'm going to talk about the structure, function, and significance of this enzyme.
And I will be going over a research article that went over the binding cleft of enzo-1,4-xylanase with 3 types of inhibitors.
So, to begin, I'm going to talk about its structure. And to start off with, it has 223 amino acid residues found within this enzyme. The N-terminus, which is actually amino acid number 34, is 1-pyroglutamic acid. Its C-terminus is serine. And it has one functional domain found within it. Within this domain, there are 13 beta strands and 1 alpha helix. The beta strands make up an anti-parallel beta sheet with beta terms included. Okay? So here's the amino acid sequence. As I said before, right here ends up being the actual start. This is part of the signaling set for transcription and translation, and it ends down here, of course, at number 223. So with post-transcriptional modification, I've mentioned already that there's a cut between number 33 and 34 amino acids to activate this enzyme. It is also glycosylated at amino acids 71, 94, and 130. This activates the enzyme further. So here is a 3D drawing of its structure. As you can see, here is the anti-parallel sheets, and here's the alpha helix, and with all the turns involved. The active site is in here, where you can see this yellow chain involved, and as well you can see the anti-parallel beta sheets and the alpha helix. So to go into its active site a little bit more, you can see all of these tyrosines and tryptophans involved, as well as prolines, which are all ringed amino acids. They have a carbon ring at the end, which helps form its binding pocket, which kind of fits with the idea of having its substrate as a sugar. Its active amino acids within catalysis are glutamic acids that work in a hydrolysis manner to cleave bonds within the xylan carbohydrate. So now moving on to its function, I just mentioned it a little bit, but it breaks down xylan, which is a component within hemicellulose, and it is the second biggest carbon reserve in nature, macromolecule-wise, behind cellulose. And the endo-1,4-xylanase actually cleaves the beta bonds within the xylanase to create xylose subunits. And it does it, as I said before, through a hydrolysis mechanism. And interestingly enough, endo-1,4-xylanase is actually secreted within or outside of cells, in extracellular areas to break down cell wall, which leads to its uses. Its uses are mainly in digestion of plant matter as its big function, because it breaks down hemicellulose, which is also found in cell walls like cellulose. So where is it found? It's found in many microorganisms. They're the natural producer of it in nature. And actually it's found in a lot of animals, but it's found in animals because these microorganisms are living within their gut. So this is how, for example, termites are able to break down wood, is because they have microorganisms that excrete these enzymes. And this is how the microorganisms themselves digest plant matter down to gain carbon and energy from it. Also, it has commercial uses now. In the paper industry, it breaks down plant matter to form pulp that's used to create paper that we use in everyday life. It also is used in animal feed, especially in chicken and bovine feed, where it's used to break down plant matter so that the animals can more easily digest it. So that way it's not taking the animal a long time to digest it, or they're not just excreting it out because they can't. So the significance of this enzyme is that it's 190 amino acids long, and it breaks down the beta bonds between xylan to form xylose subunits that are more easily digested in nature. And it has 2 major uses. The commercial uses are in the paper industry and the animal food industry that allow for greater production of these things, and its digestive uses allow insects like termites or other animals that contain these microorganisms to break down the xylan and hemicellulose present to allow them to have energy and carbon stores. So with this ends my presentation, and I'm going to pass it on to Cassie.
So as I said before, I'm going to discuss the research portion of our project. The research paper that I chose is called Covalent Binding of 3-Epoxy Alkyl Xylosides to the Active Site of Endo-1,4-Xylanase 2 from Trichoderma ricci. This paper basically wanted to see how inhibitors would bind to Endo-1,4-Xylanase 2 to determine uses in the paper industry, as Paul said, and then also for animal feed. They also wanted to determine if there was a mechanism that could be used to reverse the process for making these inhibitors. However, they did not find that there was a process like that. So in order to create the inhibitors, they first had to purify them from xylopyranoses, which are a type of carbohydrate that's full of the diastereomers used to create the inhibitors. The carbohydrate went through a Knowings condensation, where the halide on the glycoside is replaced with a oxygen, which is then methylated to create a different type of glycoside, which can allow it to be bound to other types of molecules stronger than with a halide. The carbohydrates were then taken through short layer chromatography to separate them into their different forms. So there's 3 different types of inhibitors that were used. So this is where they were separated into their 3 different types. Thin layer chromatography was used to then separate them and purify them a little bit more than they already were through the short layer chromatography. Once this was done, the carbohydrates were then treated with an alcohol, and they were then turned into benzolates so they could be stored overnight, or for however long as the researchers needed until they needed to use them in the experiment. In order to reactivate the inhibitors to, I guess, debenzolate them, they were treated with methanol, and then they were analyzed by H1 and C13 NMR to determine how pure they were and to also see how much yield was produced. The researchers found that a lot of the impurities found in the inhibitors were benzoic acid. The researchers determined that if they were to purify the inhibitors any further, they would not have enough to actually perform the experiment. So instead of purifying them further, they just decided to leave it and that they would continue as is. So next, the Endo-1,4-Xylanase 2 native crystals were purified and formed into crystals. And then they were prepared in a soaking solution of the inhibitors, which contained the water that the inhibitors were stored in, and then also ammonium sulfate. So then, once the soaking solution was prepared, the native xylanase crystals were placed in that solution and soaked overnight in order for the inhibitor to bind to the active site of the xylanase. After this was done, the native crystals in the inhibitor soaking solution were analyzed using crystallography. An R-Axis 2C crystallographer was used with a Rikugu RU200 HB rotating anode to produce an oscillating type of frequency to analyze the diffraction intensities between the bonds. So this experiment produced some pretty significant results that were kind of expected by the researchers. They determined that the inhibitors bound to the beta subunit of the xylanase. While it is all one unit, they looked at the electron density maps and determined that visually it looked like there were 2 subunits, so they decided that the beta subunit, which contained the active site, is where the inhibitors were bound. The first carbohydrate inhibitor, 4,5-epoxy pentyl beta-D-xyloside, was analyzed. The second carbohydrate, 3,4-epoxybutyl beta-D-xyloside, was then analyzed, and the bond angles between the carbohydrate and the enzyme were analyzed to see how much distortion there was from the original enzyme. And then finally, the 2,3-epoxy propyl beta-D-xyloside was also analyzed to look at these bond angles. They also looked at changes in the enzyme structure to see how the enzyme itself changed overall once it was bound to the inhibitor. So this is a depiction of the 4,5-epoxy pentyl beta-D-xyloside attached to the endo-1,4-xylanase 2. As you can see, I have labeled all of the amino acid residues that were involved in this bonding. Of course, you have the normal glutamic acid residues at 86 and 177, as Paul mentioned in his earlier analysis of the structure. However, tyrosine at 73, 88, 77, and 171 were also involved in binding. And all of these bonds were hydrogen bonds, which would make it really hard for this inhibitor to detach from the enzyme itself. This is a depiction of the 3,4-epoxybutyl beta-D-xyloside. As you can see, the two glutamic acid residues are actually covalently bonded to the carbohydrate inhibitor. And then these tyrosine residues at 88, 77, and 171 are once again hydrogen bonded to the inhibitor. Like I said before, this would make it significantly harder for the inhibitor to break away. However, these two covalent bonds would make it especially hard since they are stronger than the hydrogen bonds. And then this is a picture of the 2,3-epoxypropyl beta-D-xyloside. All of these, this glutamic acid residue at 86 is the only one that is covalently bound. The rest of the amino acid residues, the glutamic acid at 177, and then the tyrosines at 88, 77, and 177 are hydrogen bonded. So compared to the other two, this one would be a little bit stronger than the butyl xyloside. However, it would be less strong than the pentyl xyloside. And finally, when they were analyzing the changes in enzyme structure, they found that this structure right here would have had the biggest change in angstroms of the angles in the actual enzyme itself. So once the inhibitor was attached, it would have opened up more than the pentyl and the butyl xylosides. And these are our references for our project. If you guys have any questions or comments, feel free to leave them in the comment section below and we'll answer them for you. Thank you.